Hello, everyone. So first, disclaimer, I am working as a researcher and software engineer at Brain Innovation and collaborating with several MRI projects within Maastricht University. So just to let you know, I'm in the industry, basically, on paper. Okay, first, setting the scene. We are going to talk about cortical gray matter segmentation at high resolution MRI. And by high resolution MRI, I mean sub-millimeter for sure, and my current interest is even going lower, 0.3, 0.4 millimeter isotropic resolution data. And while doing this, so now I'm planning to give it like 10 to 15 minutes long, like short presentation to kind of put you in context and how I am like seeing things in this domain, cortical gray matter segmentation, high resolution MRI. So I will try to tread a fine line between not selling snake oil and also not like hiding you some secret recipes from you. Because I think I have suffered a bit during my PhD that like you go to a conference, someone says, oh, just use this software, it solves everything, segments perfectly. Then you go back, try it, and you see the like biases and errors in it. You got upset, you go to another conference, someone else says, oh, like forget about that software, use that one. And I have suffered from this a bit, so I will try to like really try to tell you what it is and how I am thinking about going about segmenting high resolution MRI data. And I assume that you watched at least partially the video that I made, that Renzo introduced a few minutes ago. So first, I would like to remind all of you where we are all coming from, maybe like going back a bit too much, and see an advice from the first neuroscientist, Santiago Ramon y Cajal. And I was actually looking at this book, The Beautiful Brain, Drawings of Cajal, just to get inspired for this talk a bit. And I saw this nice paragraph, which basically says that, like it paraphrases from what Cajal wrote in his books. Cajal says that it feels like every day you go to a forest with billion trees and you just see like in a blurry way the branches of these trees and trunks every day. And your task is to, after looking at many pictures of these trees and being inside the forest, you try to come up with a guide so that people can find their way around in this forest to describe like useful stuff, useful pathways in this forest. And he says that this is almost impossible to do if you are only drawing what you are seeing every day, looking at these pictures and then just like hyper focusing on those pictures and drawing on it. And he says that what you should do to create a useful guide is actually create a mental model of the forest without like committing too much into like singular samples, and then see what when each sample comes, like you are looking at a new picture, in his case, microscopy images, see how it fits to your mental model. If it doesn't fit, like, do you need to change your mental model? Or if it fits, it's like another evidence that your mental model is correct. So it requires a lot of thinking. Like, so this act of drawing and the place of it in neuroscience is not just like looking at a simple 2D image and drawing something on it. And of course, the relevance for us right now is that, yeah, segmentation is like this too. We look at brain images and through like algorithms or by ourselves manually, we try to just draw on it. And basically, Cajal says that you like think more than you draw and create a mental model. So now I'm going to give you 4 principles, or maybe 3, I might have dropped one. So this follows from Cajal, the principle of spatial reasoning. So you might remember the Plato's cave, where people look at shadows on the cave that is light by a light source, and then they like all their world is just to see those shadows, and they try to make sense of these shadows. And basically, what happens is that, yeah, if all you see is shadows, you will like overfit everything, all your model to the shadows. But there is also outside the things under the sunshine that you can reach. Plato says, and I think afterwards Socrates says, through reasoning. And finally, there's a nice parallel here, which is the shadows for us are the brain images that we acquire with MRI, T1 weighted images, T2 star, proton density, and we try to understand the nature of the brain. And of course, you can see from this photo from a few years ago, the neuroscientist's job is to look at the screen, try to make sense of how the brain works. So, okay, this was highlighting the importance of spatial reasoning. So the second principle that I really make use of going about segmenting like challenging high resolution data is the principle of scale space. So this is a very influential idea in computer science, in image processing particularly, which says that when you measure things, for instance, density, one might think that, oh, density is one of the truest things you can measure, like what is the density of gold versus silver, and you can distinguish just measuring the density, which one is which. However, let's consider this example. For instance, what is a cloud? Well, we all know what is a cloud, right? You can see it, you can kind of describe it or draw it. However, when you try to define quantitatively what is a cloud, some textbook definitions say that it is a certain ratio of water droplets in a 1 meter cube of air. And it looks like fine, and you might say, okay, this is an easy definition that I can make use of. However, now imagine where you zoom into individual molecules and atoms, like in the left case, and ask, like, where is the cloud? If your sampling window is so small, that ratio that you use to define a cloud given for a 1 millimeter cube of air will never work. You will either find like a complete like water, let's say. I mean, of course, you are looking at a very small pixel or cube, or just air. There will be no cloud. There is no ratio. Or if you go too much above, like beyond macroscopic scale, again, you will lose the definition of clouds too. So this just is given here to highlight that when we define things and think about objects and segments and labels, we are like these things only exist for a specific range of spatial scales. And we should be thinking about that. There is not like a true scale that is defined, gives you the true measurement of something. Like there is no true measurement of a cloud. Like the measurement of a cloud breaks down when you go too high or too low. And for me, this is similar to T1 weighted or T2 star images too. We talk about quantitative MRI, like gray matters T1 value, T2 star value, right? But if we were to do really, really high resolution MRI, maybe these like concepts will break down and we will be seeing the constituents of the gray matter, like glial cells and like MRI signal from those things, vessels and so forth. So there will not be gray matter. So we should think of this scale space. And the third principle is that I'm going to give like more practical examples a bit later, is the idea of going through this scale space through diffusion processes. And there is a very like influential work by Joachim Weikert, 1998. He wrote about nonlinear anisotropic diffusion processes and how you can implement some algorithms that make use of these concepts. And for instance, here he shows that starting from the same MRI image, by like using these diffusion ideas and maybe like tweaking how the diffusion happens like slightly differently, you can basically travel through the scale space, which means that, for instance, we all traveled in the scale space, which is when you apply Gaussian smoothing. We apply Gaussian smoothing like many times, 100 million times, and you will just get gray. There will be no grain anywhere. So everything will be isotropically and linearly diffused. Whereas this, but diffusion is more than that. You can make it nonlinear, you can make it anisotropic to preserve some structures more than others. And this is what I mean by saying traversing the scale space through diffusion. And the last principle is the structure recognition. And so by here, I mean structures in 3D. Like for instance, you can have specks that are like just one dot in a discrete lattice, like you sampled a scalar field, just like dots around. It can be like thermal noise. Or you can have surfaces like white matter, gray matter boundary, gray matter CSF boundary. This is kind of a surface, and first one was a speck. And then you can have tubes like vessels that are basically not changing along one direction, but changing in the other two directions. And finally, this structure in space can be recognized by making use of like local gradients, like how the numbers, the scalar field is changing in a given neighborhood. Like now I'm like compressing quite a bit, actually. Maybe it doesn't work, but yeah. But all I mean is that we have objects, they have like different structures in them. Some of them are like surface-like, other ones are tube-like, other ones are ball-like, and so forth. And maybe we should consider those structures inside differently. So what do I mean by all these things and how these principles come together? So now imagine this sketch that I made a few hours ago, actually. And now think of our starting point. So this is the data that we acquired at the scanner. And if we just diffuse everything, we will end up with like complete everything gray. However, maybe there are different pathways in this scale space. We start from here and we are going through the roughest, coarsest scale space, that everything is the same. We can draw different paths. And actually these paths can be drawn through tweaking different diffusion processes. And even more than that, so paths are for diffusion processes. Even more than that, each path, maybe we can tweak our diffusion algorithm in such a way that each path prefers a different structure. For instance, maybe this path, this direct path from the starting point to the end is for like isotropic cases, like wherever we have a lot of thermal noise, so that it just diffuses around and then gets straightened out. But maybe this path, the starting point becomes a bit more structured before it loses its structure, can be for cortical gray matter. And maybe there can be some other funny paths. But basically, what I would like to highlight here is that we can implement this idea of traveling through the scale space by means of diffusion, and then diffusing different structures differently in a given sample or a scalar field, as I like to think about it. And imagine that if we find like a sweet spot of the diffusion, Tn, so this is T0 time point 0, and this is the end time point. And imagine that there's an end time point in this like complicated diffusion process that maybe like the gray matter is the most structured, while everything else is less structured, so that it is like easier for us to handle and segment and see stuff. And indeed, in the video that I have made, I was making use of this approach, nonlinear anisotropic diffusion, to basically reduce a bit the noise level while enhancing the sulci in the 0.8 millimeter VASO data, T1 weighted image, basically. And this was giving me a good boost when I'm going to see the structures in the space. So, so it's all like applied. So now I'm going to give you a few like examples to show to what end do I make use of these principles. So for instance, here you can see a 0.35 millimeter MP2 range data, I think the uni image. From one run, it was like 10 minutes long from 70. And normally I acquire lots of runs to like squeeze the noise out of the data. But here I applied nonlinear anisotropic diffusion. And you can maybe appreciate that with one run, by applying the right type of diffusion, I can increase quite a bit my image quality for segmentation purposes, of course. And maybe some of you might be able to see the stria of Gennari here kind of appearing slowly. And there is another use case. For instance, here on the right side, you can see that, like, so this was a brain in a jar, basically, that stayed in a lab for long years, and it developed these chemical artifacts that are these dark spots in the white matter and partially gray matter. But these were like balls, specks, like a honeycomb pattern almost. And my task within this project was to segment this brain. And of course, those specs were giving me trouble in terms of disturbing my statistical properties in the data. And again, I used this idea, like going through the scale space by diffusion processes to smooth out these balls, these dark spots, while keeping the rest of the structures more structured. And you can appreciate, I hope, through Zoom, I don't know how good it looks, but it's working out. Like it's kind of recovering the nice image from the data without those artifacts. Of course, it all becomes easier to segment. So just a few pointers now. I'm going to go quickly. I make use of all these principles in my most recent paper that we acquired 0.35 millimeter T2 star data and MP2 range T1 images too. And basically, like the idea in this paper partially is to present a data set. It's not maybe so novel. There were people who did like a really high resolution anatomical imaging, but the rest was exciting, which is trying to give a different perspective to this data by like really highlighting the cortical landscape and like really finding ways to kind of get these folded cortices flattened and with all the depths together and finding different ways of visualizing to see what we can kind of inspect inside the data. And it goes back to Cahal's first principle in a way. Like now, by making like through like computers and all the diffusion process and like through some algorithms, we can kind of project and maybe create like more informative shadows of what is there, and like just maybe flattening it in this special way and then looking at the data might give us a bit more insight. And the one insight I got from here is that when I flattened these images in this way, preserving the high resolution of the data, I started seeing the penetrating vessels inside the data. And those actually, although spatial scales are different, do match with what Duvernoy was showing. So this is like bird's eye view of the cortex, flattened pancake. I look at it from above and I see those dark T2 star spots. And this is just the one like way that I made use of this way of thinking and then getting a new insight from my data. These are some other examples. Here you see like the chunk being flattened so that it gives us a maybe more easier to understand perspective by accounting for the curvature. You might be interested in this middle animation, maybe a bit hard to understand, but basically many of you, I think, are interested in defining like columns or parameterizing the cortex basically. But now by doing some Laney trickery, all these are contributed to Laney, of course, I can parameterize across many spatial scales, like the voxels inside my folded cortex. And you don't need to create like mutually exclusive clusters to analyze your data, but you can literally parameterize the whole cortex, fold it, and then search through whatever you are searching through across different spatial scales in the folded cortex, fully parameterized. And this is some like cake plots that show you what is on top of the cortical surface. Like for instance, here there's a Y-shaped pile vein lying on top, this dark impression. And when I push the cake layers up, I can see the cortical layers inside. And I just would like to one day be able to show it with fMRI data, and we are still in the works to do it. It's just like a data acquisition problem. It's not a big problem. Anyway, I would like to maybe skip a few. And here I can tell you that also I used all these methods not only for like one specific data, but in many different data sets. And what I am kind of showing you and making use of in that video that I recorded for you is like the tools and techniques that are tested in a very rough terrain in the last years. I segmented dolphin brain. I segmented exotic primate MRI data, postmortem data from '94, postmortem data from like yesterday, big brain data set and brainstem structure. So like basically this type of thinking, those principles that I talked to you about, they are tested and they work. And which brings me to my latest slide. So I have a proposal for you. Give me a segmentation challenge, like you, and this is kind of inspired by Andrew and Sam's data. If you have some data set that you would like to analyze, but you are struggling to find like a good ways of finding segmentations and making use, like highlighting the laminar structure inside or whatever, columnar. Yeah, let's get in touch and show me that data, and I will prove, I am quite confident that by using these techniques that I talk about, I will be able to segment them in a reasonable time. And by that, we can switch to the questions and answers session.
How much do you charge for segmentation assistance?
First, send an email.
Joking.
But zero for now. Maybe I should charge actually.
I mean, you work for Brain Voyagers, so.
Yeah. I mean, I am very passionate about this, so I am not like money focused about this part. I would like to really, like, I am very excited by high resolution data. I want to just record and like highlight like all the unique features of these like beautiful images that we acquire at ultra high field. And I feel that there is kind of a gap between like MRI physicists acquiring like really nice high resolution images versus like neuroscientists trying to analyze them. And like, for instance, when I published my recent paper, this 0.35 millimeter T2 star dataset, I had a lot of struggle because my feeling was that MRI physics in general, I mean, it's a generalization, of course, we're like thinking that, yeah, what is new here? Like people did it like 10 years ago, 0.35 millimeter isotropic T2 star weighted images, whatever. Like, what did you do? Well, my novelty there was not only acquiring the data, but also like analyzing it such a level that it is like very useful for neuroscientists, and it produces the framework and the basis of future analysis. And of course, Renzo knows this. Like, I contributed all these pipeline developments to Laney between 2019 and until the paper is published in 2022. Basically, it's like a 3 years of tool development. Anyway, I'm like drawing a tangent now.
Faruk, I have a question. So from last year's OHBM, you walked me through like the algorithms, and I really find these fascinating and so useful and interesting. And I had a question a little bit about some of the noise. So I was wondering if you could go back to your slides, the one where you're showing a primate photo. Yeah, that one. So this image here. So I like the one thing I have is as the noise gets more aggressive, let's say, or the spots. So I want to. Well, I don't know. I just have a question for your opinion. So where is the hardest part of these things? Because the idea of using diffusion and kind of anisotropic smoothing and that I think is quite good. And I agree that basically what we're trying to do is segment based on a scalar image. So one thing is having diffusion tensor imaging is another way to go with this. And that often really helps delineate things. And that might be something else to consider, non-scalar images for boosting contrast, and either multiple contrast in your anatomical, like you have the inversion 1, 2, and uni, that kind of thing.
Yeah,
that's kind of a little vector to help you segment. Another thing is diffusion tensor imaging certainly helps segment even the boundaries of gray and white. So here on the right side of this image, maybe the third gyrus up from the bottom on the right. Yeah, right where your mouse is, a little northeast of there, more lateral. Yeah, so there are a couple very dark spots in that in the next yeah, there. So actually, if you look and see what the algorithm does, eventually the noise gets so big that at boundaries now it becomes, I would say, confused a little bit, right? Because there probably shouldn't be a divot out of there. It should be smooth going across. And I just wonder, how do you put error bars on what you're doing, or how do you really check for what strikes me as using the information of like the linear progression of it, because now the gray matter is going across and now there's like a chunk knocked out of it and then it goes across because of this. So how do you deal with these things?
Yeah, so like I had similar discussions many times. Basically, whoever looked at this diffusion GIFs made a similar comment. So basically, like just looking at this 2D slice and saying that, oh, there the algorithm is making a mistake and creating from this darkness gray matter. Like to make that statement is similar to just living in the Plato's cave. Like you are looking at just a single slice in 2D. You do not know what is below and above of this slice, right? But the algorithm knows, because this is completely 3D. So I mean, I do not remember exactly what was the case in this data, but in many cases where I like had like got criticisms about, oh, like here the algorithm is doing the wrong thing. When I went back and see the slices above and below, it was actually not the wrong thing. It was the sensible thing to do. However, having said this, it also doesn't mean that the algorithm doesn't create biases and artifacts, right? I mean, at the end, this is kind of a smoothing diffusion. Like we are making up stuff based on some principles, and actually we are baking our like thoughts and assumptions through this like algorithm into the data. And of course, like one should always be careful about this. And what I actually do in practice is that I smooth the images. I kind of get a nice initial segmentation out of it, but I always do quality control, like thoroughly, like going through slice by slice and loading both the smoothed and the unsmoothed image. And when I'm in doubt, I check, okay, how this local was looking in the unsmoothed version. So basically, you cannot just drop your original image and just smooth and say, oh, this is my new image and it's the truth. No, like you need to make sense of both. There was another part of the question which was making use of the multiple modalities. Yeah, I agree with this completely. If I had the chance, actually, more than maybe like denoising or smoothing, I would prefer to get different contrasts that are sensitive to different macromolecular compositions of the tissue. And for me, like, yeah, getting T1 and T2 star in the living brains is, for instance, like both extremely useful. You cannot just rely on T1, you cannot just rely on T2 star to detect like different types of tissues. You need both. So about this multi-modal perspective, I agree with you completely that it's like useful in general.
And to be clear, I mean, just because maybe there's one dot here that I would question or something, I mean, the algorithm is great. I guess, it's going to be bad no matter what. So maybe this one isn't fixed. I guess I'm wondering a little bit, can you put a bit of a probability on these things as well? So for example, can your algorithm know I had a hard time with this one, or I was least sure with this one? So that would also help you with QC, because now, again, we know and love quality control, but it also takes a huge amount of time. And I think, people want to, I don't know, anyways, can you have kind of a probability or something come out that points at problem areas?
Yeah, I did not work on it specifically in depth, but I had some ideas and discussions about implementing some like entropy metrics to figure out like what type of structuredness you start from and what type of structuredness you end up with, but not in too much depth. But also like those metrics have their own biases too. So it is very tricky because you can implement like something that tells you like probability or whatever, like how good the algorithm did there. But you should be aware of at the same time the advantages and drawbacks of these metrics too. So I think even with those things, like you need multiple measurements to figure out what's going on.
Sure. Yeah, I mean, at some point, but you got to make a decision. These are things that kind of help, basically shrinking the number of problem points down and down. And it's true that you might be, kind of like ChatGPT or something. You may be just like so sure of yourself at the worst possible moment. That kind of thing may happen too. But anyway, I really think these are impressive, and I think it helps quite a lot.
Oh, and also like one point I forgot to mention, like I am not obsessed about like doing manual segmentations. I don't want to do manual segmentations. I don't want to think about like which filter to run and which like whatever. Like, no, I don't want to do it. Like I want a big red button. I give the data, I press it, and it gives me the perfect segmentation. However, it is like my problem is that this is not happening. I tried a lot of tools like FreeSurfer, SPM, Brain Voyager, CBS tools, now NIRES, like they have their preferred type of data. And like for me, the rule is that, okay, like look at which lab produced that software and then try to acquire data exactly as that lab is acquiring, then things will work. But the moment you start like dealing with different types of data, for instance, like segmenting vaso images, in the EPI readout, distorted a bit with like a funny outside and so forth, like then all of a sudden you are kind of there, like looking at the data without anything to do. And then you revert to the simplest thing, which is like slice by slice going and trying to draw. My point here is that there are cases where we need to just go slice by slice and draw and spend hours doing it. But maybe there's also like a somewhere in the middle where we are trying to find these like optimal strategies and techniques to apply to these new types of data, like operating on some principles to derive and filter out what works and what doesn't. And whatever stands the test of time, and for me, what I talked about were the tools that stand the test of time, should be the ones that become like more automatic in the future for like mass production, whatever.
Maybe to add from my side, like one thing that I learned from your video and from watching you segment is, aside of all the principles you mentioned, the interactiveness, right? In Segmentator, you kind of draw in the histogram of gradient versus the signal magnitude, and then you see it directly. And also in your kind of your entire setup, right, of the tablet is to kind of have the interactions in there. So one thing, like I started with drawings and doing segmentation slice by slice, but as you said, with the Cajal principle, kind of imagining the sulcus in your mind you do it not slice by slice, but you do it kind of sulcus by sulcus, right? Based on a structure that you imagine. And it was a bit maybe annoying watching you do the segmentation, because you always have this interactive loop, right? You go through slices and it flickers and whatever, but I think that was the most important message for me, that the interactiveness, because these tools are great, but you kind of need to tweak them as you see the data. So I think getting the feedback loop short is the tricky part.
Yeah, and it's all about, again, Plato's cave. Like, we are looking at these shadows, and we need to, like, I mean, we just have like 2D in a way, like two eyes, that light comes and creates this image. If we were to live in a 4D or whatever and see like 3D by its totality, then it would be easy for us to, I guess, understand the curvature of the brain and so forth. But we cannot do it. Therefore, we need to find ways, like quick ways, to kind of approximate to that type of understanding. Yeah, I don't know. I'm maybe a bit too abstract now.
Well, someone would ask you to segment in 4D or high. Someone will always ask you for the next hardest thing. That's
yes.
That's the rule of software.
I actually implemented the nonlinear diffusion filter for 4D, and it does work when you make up data.
Yeah, wants to apply twister theory to the brain, so I guess that's how you would parcelate that.
So what do you think about these machine learning based segmentation tools, which I think are sometimes quite impressive? Like, you need to have a training data set. Or Brain Voyager, they had this tiramisu thing, which I think works for, I'm not sure if it's used a lot, but do you think that maybe like our huge amount of time that we spend on manual segmentation, there's like room for optimism with machine learning based approaches, or how generalizable can they be? Like, if I change my VASO protocol from one parameter to another one, would it still work? Like with the tiramisu, it doesn't seem to, or...
Yeah, so disclaimer, personally, I do not find machine learning interesting. So I do not like read about it. I do not think about it too much. So I am like a naive about it. However, like based on the results, and of course, like this always comes in conferences. So like there's this machine learning thing that solves the issue, whatever. The thing is that like making any algorithm create something that looks good from a distance is quite easy. Like, also you can just cherry pick like which slice you are showing, but like conveying like what it did in the whole 3D whole brain image is like, this is very hard. You cannot convey it in a PDF. Basically, you need to be able to download the data, download the segmentation, and you need to go through the slices, look at it, and see where are the like errors in as a result. And basically, for anyone to judge these any solution, I would recommend like you have to see the data and the results in 3D and spend time on it, and then make up your mind, and do not like jump into conclusion that, oh, like as a poster it shows everything nice, like it solved the issue. No. And the other danger is that sometimes, I mean, of course, people don't have like negative aims while doing it. Like sometimes, for instance, you just don't know what is the sagittal sinus, like where is it, and how does it look like in a T1 weighted image? And you might, like, both of us can look at the same image with the same segmentation, and one of us can say, oh, this is perfectly segmented, whereas the other one, who knows a bit more about the neuroanatomy, can say, this is completely wrong. You are getting like 3 millimeters into the sagittal sinus. What are you doing? You are looking for like PRF maps in the sagittal sinus, So this is another like complication. Like, we need to train our mind's eye too by looking at some medical textbooks, studying a bit of neuroanatomy, where the main like veins, arteries are, sinuses are, and then interpreting the this is also what I mean and associate with like Cahal's thinking, like having this mental model a bit, and not just looking at the gray values of each pixel to say this is gray matter or white matter.
You also have to remember that there will be non-standard physiology in case surgical
Indeed,
patients. Also, I mean, the variety of cases is truly astounding, and those are the ones that people are often interested in, right? They're kind of doing this as a proof of concept on healthy, and now you want something where there's cortical thinning or white matter lesions and things like that. So, that, to me, that's where the machine learning things really get tricky, because of the limits that people want to push things to.
Yeah, and also I would like to see more in the literature and also in my work, in the future work, I would like to highlight the edge cases. I think they are like incredibly valuable. Rather than showing, hey, it works. I mean, yeah, you show, hey, it works, but also show me like when it doesn't work. Because knowing these algorithmic pathologies are so important for us to be able to like move further. Otherwise, like every user has to figure out by themselves. Or like in this case, if you acquire MP2 data with this, whatever, with this parameter, like it overestimates the motor cortex. Yeah, if you have to figure it out by yourself, it's like a lot of work to do. Oh, and also there's another point about this machine learning stuff. Like someone has to, well, I am not up to date too much, but I believe there is a value, an immense value on creating validation data sets, like for judging how the algorithm did, right? And how do we create these data sets? Well, basically you have experts looking at the data like the beginnings of machine learning and labeling, oh, there's a motorcycle, there's a bus, there's a cat, whatever. We have to do it for the MRI data too. So this careful manual work for a given time period, and depending on how many people are doing it, this can take a decade or like 100 years, I guess. Creating that kind of like a benchmark data set on different MRI contrasts and different scanners and whatever, I think will be very useful anyway. So I think we even using like machine learning methods at the end, we cannot avoid doing some manual work to create these validation data sets.
I think that would be useful even to incorporate within a study. I mean, if you have 5 brains in an experiment, it would be nice to have some sense of inter-segmenter reliability. You have 2 different people segment and report the overlap. And the problem is you have no measure of your error.
Yeah. But
it's if you think it's good, it's good.
Well, I mean, we have to base our algorithms on something. And also, this is another kind of thinking. Maybe I'm stretching a bit what Eli is saying, but I encountered this thinking, like saying, oh, I use an automatic algorithm, therefore it is better than doing it manually. Like, no, automatic algorithm is someone thinking and having like his own ideas and assumptions and baking them into a computer so that computer can do it, right? And there can be hidden assumptions and implicit like biases and so forth. And you are basically taking all of that, and just because it's automatic, you think it's like better. No, it's not better. Like you have to be able to judge. And like, what do we do? Like, I mean, at the end, I guess we are trying to replicate the human vision and human understanding. And therefore we have to base our error bars on with regards to what humans are capable of right now.
I mean, we face this issue in retinotopy. So you do a retinotopic mapping experiment and you want to draw boundaries between V1, V2, and V3. People talk about it as if it's easy, but the truth is 5 people will draw those boundaries in different locations.
Oh, okay. So this actually is a good point that I would like to talk about a bit. Like we need to set an error. Are we expecting infinite precision across 5 people to determine where the V1, V2 boundary is? Or maybe it, like if it is up to 3 millimeters different, it doesn't matter because it is somewhere there, a bit like the cloud argument. Like there is not like a one finitely sharp scale that the definition of cloud breaks down. No, it's like a blurry. So I think like some level of error in our measurements, and also this is like a very old engineering concept, having tolerances when you are building any machine. Like you cannot build a machine with 0 tolerance, then it will not work, basically.
Yeah, of course, error's part of the game, but it'd be nice to know what the error is, estimate the error. And then for layer-fMRI, the tolerance is actually pretty small, right? Because the cortical ribbon is so thin.
Yeah, I would
for,
say.
Yeah, I was gonna-
defining area boundaries, you have millimeters, but you don't really have that much leeway for layer-fMRI.
Tyler, you were saying something?
Yeah, I was just going to add to that. I mean, it's a complicated point because oftentimes we're working with processes where we actually don't know what the scale is. We're trying to discover the scale at the same time that we're trying to define it. And so especially, I mean, you were saying 3 millimeters is not a big deal for retinotopy, but for a vision scientist, there are often cases where that's a huge deal. That's the difference between columns, obviously. And so yeah, I can imagine situations where like V1, V2 boundary is not a big deal for it to be off, but if you're trying to find the precision of that first stripe, then that would be like a big part of your study. So it's probably very different for every case, obviously.
Yeah, indeed. I guess what we are agreeing on is that like this, like saying infinite precision is always better and zero error is always better is not the case. Like there are cases where you can live with some error, and we are living with some error every day, basically. Like nothing that we use or build is built with zero error.
My point isn't to achieve
Yeah.
zero error. My point is to put an error bar on the layer definition. What is our confidence of the segmentation? I have no idea how to derive that, but it would be nice to put a bound on that somehow.
Actually, about that, maybe not an answer, but kind of a riff on it. I think we should report the effective resolution of our regions of interest. And what do I mean by that? Like we have voxels, right? And we have cortical thickness measured at a certain level. And I mean, like, for instance, like comparing a rat study and a human study and saying that, oh, rat study is at like 0.2 millimeter isotropic resolution means almost nothing. Like you need to be able to say it relative to the object that you are measuring, and maybe we can call it like relative resolution, effective resolution, whatever. And also like similarly, like 0.8 millimeter isotropic fMRI at V1 versus like frontal cortex can be very different because V1 is very thin, other areas are like thicker, and I think we should at least like kind of state, I am converging to this thought more and more every day, like to report the average thickness or effective resolution in the ROI that is being analyzed. And actually, since we know the cortical thickness changes based on sulcus and gyrus too, maybe we should report it in like 3 regimes, like gyrus, like the tip of the gyrus, and like bottom of the sulcus, and the walls in between, so that we have a kind of a nicer idea about the ROI.
Faruk had asked me to keep an eye on the chat. Paul has got something which might have been part of the previous discussion. Paul, do you want to say something about it?
Oh, well, no, just relate again, this idea that going back to the cloud thing. Actually, one time I jumped out of a plane with a parachute, and then actually kind of fell through a cloud, which you're not really supposed to do. But I wanted to go into atmospheric physics for a while, so I really, like cloud study is really hard. But this idea of there is no boundary on a cloud. Like you fall through, you kind of don't even know, except things get a little fuzzy, and then you go through. So this kind of scale argument, but these things are a continuum, so there really isn't a boundary. And in the same sense of when Linnaean classification, this is this kind of finch, that's that kind of finch, but the boundary is actually you'll drive yourself mad. It's kind of a fractal problem. Where's the boundary of Norway? Good luck. It really depends on the scale you're looking at. So again, people, when they publish stuff, they want these absolutes in that. But I do think this idea of a probability, and I guess that's kind of what I was getting at with that, looking at the gap, or what I thought might be an error, and that those are two different kinds of probability a little bit. One is I fix something, how sure am I of it, or I change something appearance. Another one is where is this boundary, and that is a blurry thing. I mean, essentially it gets to the irony of you're discretizing something and then putting back a continuum over it. But I do think the idea of error bars in any measure is useful. This is one where you're like, any kind of measurement that doesn't have it should probably be a little skeptical of.
So for example, let's say you're looking at feedback in V1, and as you get to superficial layers, the signal gets stronger. I'd like to be able to plot the probability that you're in a superficial cortical depth versus the magnitude of your effect.
So something like kind of little Monte Carlo, there's a way you could kind of feed back and say, based on what I've predicted here, I don't know if there's a way to kind of simulate something. It's a little bit hard with scalar value to picture this.
Well, it's also hard if you're doing this manually.
Yes. Well, that's right. Then now you get into self-reporting things like this, like, well, I'm a little less sure here, or something like that.
Right. Or inter-rater reliability. And if you could have 5 people segment this one brain.
It's a valuable data set.
Yeah.
Or
But that
even if
also-
like,
Yeah, Tyler, go ahead. Fine.
I was just going to add that, I mean, some of these measurements actually might be useful for us as kind of a principle. Faruk, you'll be acquainted with this, but one of the things that kind of I thought was really cool when I was learning to draw was that like when you draw eyes, most people draw like a hard border around the iris, but that doesn't actually exist. And they look very unrealistic until you start incorporating the fact that there is no boundary. It's just a smooth kind of gradient between them. And I sometimes think about like the higher resolutions that we get with some of the layer principles that we go to. Actually, there's not going to be this really strong cutoff point where it's like this is now gray and this is not. And it might be really interesting for us to actually discover the functions of that transition. So the error bars might be useful to us.
You might be able to make a distribution of all, let's say you've segmented something that you want, and you just take all those scalar values and make a distribution. And if you're on the edge of that, maybe you'd be willing to concede that that's a reasonable sense of certainty or not. I don't know. It depends how homogeneous the brightness is expected to be across something.
I would like to maybe bring the discussion to not only kind of how to solve the problem and think about it, but maybe the magnitude of the problem. So I wanted to get Faruk's opinion about, like, let's say I do a whole brain VASA study with the kind of data quality that I sent you, but I'm not interested in one sulcus, but the entire brain. And let's say I scan the hell out of it. I do 100 runs, right? 100 times 50 minutes. Will I spend more time segmenting or will I spend more time scanning? Can you give me an estimate? Like it took an hour to get one sulcus, right? Approximately in that video.
I mean, when you pose it as either or, I think I would rather not answer because, yeah, why not both? Or what prohibits you from doing both, you know? I mean, like about this spending time drawing argument, like we should remember which field we are working in. We are doing neuroscience. I mean, I hope, like most of us, or trying to do it. And the basis of neuroscience is literally people looking at microscope for years and then like drawing stuff. So if you spend like 2 days drawing like or quality controlling like thoroughly, or like removing the big errors at least, is pretty quick. Like you are not spending 1 year to write like one drawing, you know. So I think we should like kind of like we actually posed this problem in one of my first papers in the segmentator paper in 2018. Like we say, okay, like the neuroscientist, neuroimaging person can have 2 paths. Like one is I run something and live with the biases and errors that are available inside, and maybe try to do it like many times and then average and hope that it averages out the errors. Kind of like a crooked thinking, but because you cannot get rid of bias that way, you can get rid of like variance of it. And the second way is that I will look at my data and I will try to do my best until I am satisfied with the results that there are no big errors left. So you can decide, you know, and I don't think like one way is superior to the other one all the times, because we live under like time constraints, money constraints, other stuff. But I would say that if you can afford, like, why not going through the data, spending extra effort to kind of be relaxed about it later, to say that, hey, I looked thoroughly and I got rid of the big errors in my data and I quality controlled it thoroughly, and now I can believe what I'm going to report. So I think this is the axis that I would think about with regards to your question. Like if you are satisfied with the VASO whole brain data and let's say the first output you got from FreeSurfer, I mean, it's fine, but when you are interpreting your results, maybe around V1, consider that it could be the case that there was a bias and you got too much of the like sagittal sinus, and maybe there is like a misrepresentation of some signal there. But if you are trying to reach to like a conclusion about the whole brain, like everywhere included, maybe like that amount of error doesn't matter. So it is not easy to say.
I think I was more thinking of kind of the major limitations in layer-fMRI as we know it right now. I think like people talk about SNR and that we have limited sensitivity and therefore we need long scan times and so. But I think it's not unrealistic to think that if I want a whole brain segmented at the quality that you did in your video, I need at least about 100 hours of manual segmentation. So the mental energy that is spent of layer-fMRI students is like more on this manual segmentation than almost like anything else. So I think it's really a big problem in layer-fMRI to make it practical. And because it's done by students, you don't really hear about it in the conferences, and maybe PIs don't worry about it so much. But I think manual segmentation is a time sink and a mental energy sink. And it's a huge issue. So any little bit of the tricks that you mentioned
yeah, I mean, totally. And actually, like this is my current goal, at least in my free time, to create these YouTube videos and yeah, like put it out there so that it might help some people to like semi-automatically speed up some parts of this process. Because although I can do it, I don't like doing it either. Like I would rather do something else, like develop an algorithm, scan, whatever. But the issue is that like if I don't do it, I don't feel comfortable like just going ahead, analyzing my layers in whatever A1, B1, and then writing stuff about it. I have to be satisfied with the quality, because it's so important for layer-fMRI. You know, if we are going to interpret the layers, we need to put the layers correctly there. That's like a big part of the analysis. Maybe I wouldn't do whole brain layer-fMRI. I would do more like ROI specific. It's also too much data. Having said this, I am doing like 0.1 millimeter whole brain analysis right now using the big brain data set, not fMRI, but looking at something else and also using 0.35 millimeter like whole brain data. So I mean, like I'm doing it, even though I think for layer-fMRI purposes, focusing on an ROI is maybe a better approach. Paul, you were saying something.
Well, actually, this got back to a technical point. I was wondering if you use edge filters, edge-based filters, and kind of that to help you, because basically with a scalar image, you're cleaning and then you're looking for changes, and it's basically a slope that you're looking at and deciding at this point on the slope it's steep enough for me to do it. Some of those are well-posed problems. If you've cleaned, it gets a bit, all these things get circular, but
I make use of gradient magnitude quite a bit as a like edge filter. So basically you compute the rate of change in XYZ directions, and then you take the magnitude of this vector field derived by a scalar field. And that already highlights like I mean, for instance, like one thing that I see over the years, we were discussing with Renzo like yesterday, with the T2 star weighted data, like sometimes people want to like do layer-fMRI analysis, and to preserve the readout distortions and so forth, they want to like segment based on the T2 star weighted data. Like I almost never saw confidently the white matter gray matter boundary consistently across areas in a T2 star weighted image. And this I can also prove by using what you are saying, like putting a edge filter to highlight like literally like number wise what type of fluctuation is there between tissues. And at the T2 star in many areas you do not see this like edge, but if you had a T1 weighted image, you see very clearly the boundary. So the decision becomes very straightforward in that case. So in such a case, I would make use of the edge filters if I am like unsure to figure out what if there's an edge or not in the data, or how strong the edge is.
And it's a principal part in the segmentator analysis, right?
Yes, I mean, I don't know who people here are familiar or not, but yes, there's like a whole little field in image processing that makes use of these gradient images in combination with the intensity images. So it's actually funny, like there is a hidden contrast in your data, and it's always there for free, which is the computing the gradient magnitude based on your scalar image. And then like scalar image together with the gradient magnitude, both, which was the case in the segmentator data and software, like can highlight certain structures even further. And actually the diffusion filter is based on that too, because you derive a second moment matrix, which is like gradients of the gradients in a way, and then it tells you about the local structure there are these like freely available data, like derived information in your data already.
Yeah, I think that like difference of Gaussians is an approximate for the Laplacian. That's a, anyways, numerically efficient version.
Yeah.
We are at the full hour. So
Yeah, I don't know if this was useful. Like, can you tell me if this was useful or if it was just like a like conversation random?
to me,
So-
the video was awesome. Like all the little tricks to see the commands, and then yeah, like the low level like, tricks. That was.
I mean, it was not a tutorial at the end what we did today, but I hope it was useful somehow. Okay, then thank you.
All right. Thank you. Thank you.